So we've run a fairly standard, I would say, sort of SAD-MAD phase 1 study that we're now able to report out, where we did escalating single doses of SAT-3247, and then we did escalating 7 days of dosing of the compound. And we did the study, as you typically would, very much focused on safety, but also on PK, how the drug was being handled in people compared to what we'd seen in animals, and what it was going to take for us to get the appropriate exposure to drug in people that we believe is going to drive, ultimately going to drive efficacy. And there was a number of findings from the study that we're very excited about. The first was that the compound was extremely safe at all doses. Which means we definitely have the window we need over and above where we think we're going to be for efficacious doses to dose this compound going forward. And any changes we did see in the healthy volunteers reflected what we had seen as we'd gone up in doses in our animal models. And they were things that were very easily monitorable, they weren't adverse, and so everything we might have predicted we would see, we did, which isn't always the case, but that's what we definitely saw on the safety side. And then on the PK side, maybe 3 or 4 things that we were able to, data that we were able to collect, which were important to us. One was the handling of the drug in people was very dose related, which is good. So it was very predictable from that point of view, which we liked. At the doses that we believe are going to be efficacious doses, there was no accumulation of the drug. So with repeat dosing, it looked very much like it did after single dose. I think that's always important so that with longer term dosing, you don't end up getting to any toxicity issues. But also important for us in terms of maintaining the optimal coverage of the target that we need to get us to where we want to be as far as efficacy is concerned. And that was the other thing we saw there. When you move from animals initially into people, you're scaling everything to say, okay, this is where we are in the animals, this is where we think we're going to need to be in people. Once you get into the healthy volunteer studies, you can check that by saying, okay, did I get the sort of coverage of drug that I thought I would? And the answer was that the scaling actually held up very well for us in terms of the sort of doses that we were thinking we would need to get the sort of coverage on the target we think we need for efficacy. So still a step away from knowing if we've got efficacy, of course, in people, but connecting all of those dots, which is important to us. The other reason that was important was we had moved ahead so that the other differentiating piece for our drug maybe is that this is a very simple drug for patients to take. Obviously, DMD patients sometimes find it difficult to get to centers, and invasive procedures for delivering drugs and stuff can be tough, and those sorts of things. This is an orally available small molecule just in a pill form. And when we scaled where we thought our doses were going to be, that also drove us to make pills of certain dose strengths, right? Because we thought we were going to need to cover certain doses. Obviously, we didn't make huge amounts at this initial stage, but if things that had been very different for us as we translated into humans, we might have had to go back and do a little more CMC work to come up with some different dose strengths. But as it was, we're well positioned going forward as we put together where we're going next in patients and our phase 2 program and those sorts of things, with the dose strength of the pills that we have for the compound. So yeah, a very exciting initial study, showed safety just as we needed to, and showed PK that we were very excited about based on the translation from animals, and also, as I say, where we are in terms of the dose we need, which is a very, a good dose level. It's not a high dose level in terms of size of tablets or anything like that, is really giving us the coverage we think we're going to need for efficacy. There is one final piece that I won't be reporting on here, only because this meeting's just come up on us a little more quickly than, and we just don't have the data yet. But we were able to extend our phase 1 into, so we ran this initial SAD-MAD study in Australia. And in Australia, we were able to extend our phase 1 into a group of up to 10 adult DMD patients. And once again, to be completely clear, the reason we wanted to do that was to really look at safety and PK once again, mainly focused on making sure that that PK that translated into healthy volunteers for us is translating also nicely into patients. As we go forward with our phase 2 clinical program, we don't want to miss because we're giving the wrong doses, right? I mean, we're a small biotech company. We're very well aware that, you know, we don't get many chances at this. And so to be sure of where we sit with that PK, we wanted the opportunity, if possible, to actually look into some patients as well. We are collecting some, we're not collecting anything like biopsies or anything from these. These are adult DMD patients, but we will be collecting because they've been kind enough to do a 28-day study with us. This is only 28 days of dosing. We will be collecting some, monitoring some endpoints off the back of those studies. I don't think we're thinking that 28 days of dosing will be long enough of administration of the drug, so not making any claims about what we will see there, but just as full disclosure, we are collecting some other readouts. But those patients are just being recruited at the moment. I don't have data to share from those patients, but that will be our initial patient data. We hope to be able to share that with everybody by second quarter. I mean, it's soon, relatively soon, but don't have any comment to make on that right at this time.